I hope your day's going well. We're going to jump into our next lecture here within Unit 1. And in this lecture, we're going to actually revisit what we've talked about in a little bit of a different way, and in general talk about how things are organized within microbes and living things. To begin with, I want to jump back into a slide that we saw at the end of the last lecture, which was comparing different levels of organization within biological systems. And if we're thinking about organisms, we talked about pretty extensively in the last lecture cells. And cells are the smallest fundamental unit of life. We mentioned this back in the very first lecture. We took a more specific look at cells and the different types of cells in the last lecture. So this isn't something that's new to the conversation that we've been having. In the last lecture, we also broke it down to a bit of a smaller level and focused on cell structures. So different types of cells, eukaryotes and prokaryotes. What do the cell structures, the different organized regions of each cell look like? What functions do they carry out? Those smaller structures go into making up the cell as a whole. And so we identify those as the functional units of the cell, the smaller pieces or smaller regions, smaller organizations that make up the cell overall to carry out processes of life. In this lecture, we are also going to visit molecules. And we mentioned molecules briefly last time with a focus on macromolecules, which will again be our focus in this lecture. But molecules are the building blocks that go into making those cell structures. So we're going to an even smaller level. We have to be able to think about what are the functional pieces that go into building the bigger organizations, the bigger regions of a cell that go into building the cell overall. And then we mentioned that there was a term virulence factor that we wanted to be familiar with, which is a structure that has some function specifically related to infection, either helping a microbe to colonize or to avoid an immune response when it is carrying out infection, and that we should always be focused on, is this a virulence factor? If so, what is its virulence action, so that we can characterize these different parts in terms of what they're doing and how it's related to health. Keep in mind, virulence factors will be cell structures or molecules. So it's not a full cell, it's not a full pathogen. It's a part that's carrying out a specific role that's assisting in causing disease. Okay, so if we think about microbes and the cells that make up microbes, and most specifically when we talk about bacteria, which will be one of the primary microbes that we talk about in this course, one of the things that's helpful when thinking about these cells, when thinking about these fundamental structures, is being able to describe them. And the reason why it's helpful to be able to describe characteristics about cells is because it allows us to determine what species of bacteria we're looking at. So when we are thinking about bacterial cells, it's important to keep in mind that bacteria are made up of just one cell. So the entire organism is one cell. And if we look at how those cells are shaped and organized in terms of arranging with each other or grouping with each other, we get some characteristics about a specific species. For example, E. coli will look different than the species Staphylococcus aureus. Both of those have certain strains that can cause issues in health, but they are very different organisms. They're different species carrying out different processes that lead to different diseases. And so fundamentally for us, it's important to be able to describe them, and shape and arrangement are really, really effective ways to do that. Okay, the first thing I want to focus on is the shape of the cell that we may see when we're looking at bacteria. And there are multiple different shapes that bacteria can take on. We will focus on two of these, but I want to highlight multiple shapes that we may see. The two primary shapes that we will see are what are known as cocci and bacilli. However, I want you to keep in mind that there are these other shapes as well, which I'll actually cover first. Vibrio, which is sort of a kidney shape, comma form, looks like a comma, this corkscrew form, a spirochete. There's a variety of different spiral-shaped cells. And you can see that we're physically we're just describing physically what these cell types look like, right? And that's helpful because now we know if we ever see that, this is potentially the type of bacteria that we are looking at. Okay, the two most common shapes that we run into are cocci and bacilli, like we mentioned. And so take a look at this diagram. All of these are cells that are cocci, and just take a second for yourself. What shape is a coccus cell, or are these cocci cells? And you probably arrived at what we're all thinking. Matt, it's pretty obvious. They're round, right? These are spheres. If we're thinking about them in three-dimensional space, they're like a ball. They're like a sphere. And so cocci describes cells that have this round shape to them. If we take a look at bacilli, how would you describe those shapes within this lower graphic? And you're probably saying, well, I may not have the perfect word to be able to describe this, but I would say that they're longer, right? And so bacilli are elongated cells. These are also known as rod-shaped. So if you ever hear rods is referring to cells that are bacilli in shape. Now, importantly, when we are describing the shape, there is a more technical term that we use known as morphology. And so you may have seen morphology used before or come across that sometime before previously in biology. But when you hear morphology, really what we're doing is we're just describing the shape of any individual cell. And we can describe a collection of cells, but we do that in a little bit of a different way that we'll talk about here in a second. Right now, what we're focused on is just the shape of any individual cell. Okay, going back to the example I used, the Staphylococcus aureus and E. coli cells, those are two different species. Staphylococcus cells are cocci in terms of their morphology. So if you were to look at them under a microscope, you would see that round shape. E. coli cells are bacilli. They're rods. Pretty obviously, if you look at those under a microscope, you will see that the cells are elongated for their morphology, rod-shaped or bacilli. Okay, so that gives us the shape of an individual cell. The other thing that we want to describe that you probably noticed is going on in this diagram is how cells are clustering with each other, or how they are grouping with each other is a better way to put it. And we describe that as the arrangement that the cells take on. You can think of it, how are they arranging themselves in terms of how they exist with each other. And you'll notice that there's a variety of different arrangements, and that in some cases there appears to be no arrangement at all. So if you take a look at this upper leftmost cell, the coccus cell, it's round. We know that its shape is coccus. Since it isn't grouped with any other cells, we would just describe this as singular. If you take a look at the cells to the right of it, you'll notice that now there are two connected to each other. And so we're going to describe that arrangement a bit differently. Rather than it being singular, there's two of them, so we describe them as diplo. And we can put diplococci together to sort of combine the morphology and arrangement. Typically, we will keep those separate, so we would describe these as diplo because there are two of them. If we go down from there, we can see that now we have cells that are all connected to each other and grouped in what appears to be a chain. And when cells arrange into a chain, they take on what is known as a strepto arrangement. If we take a look up here in the upper right, you will see that the cells are now clustering with each other. So now we have a bunch of cells together. And I do want to highlight for you that these are almost always drawn in these cartoon graphics as sort of this triangular cluster. The cluster is not triangular. That's just because it's easy to draw that way. Clustering is going to be any type of grouping where you have a bunch of cells connected to each other in this big group. And that arrangement is what we call Staphylon. Okay, then there are two more that we see here that we'll focus on. The diplo up here that's encapsulated, we'll revisit what that means in the future. But for now, we'll take a look at these two lower arrangements. And we'll actually start with the one on the right, tetrad. Tetrad is where you have 4 cells that arrange with each other. So you have this consistent grouping of 4 cells that come in contact with each other. And then there's Sarcina, which is where you have the 4 cells, but stacked with another 4 cells. So overall, you have this group of 8 cells that you can detect. Okay, and then if we take a look at bacilli, we'll notice that we see a lot of similar arrangements. With a couple of exceptions. Again, we have a cell that isn't grouped with any others. That would be a singular bacillus cell. If we have 2, the arrangement is diplo, diplo meaning 2. If we have a chain, we see a strepto arrangement. And for bacilli cells, sometimes you'll see this arrangement where they're, instead of lined up end to end where they're elongated, they sort of sit parallel to each other, almost like fence posts, and that's what's known as a palisades arrangement. Now, one thing I want to highlight here that is important to make note of is the fact that you will notice that the bacilli cells do not take on the Staphylon arrangement. Staphylon is an arrangement that we just see with those round cocci cells. And it's because of the nature of where arrangement comes from for any of these cells. The reason why some of these cells group in these very specific and unique ways is because of how they divide. When the cells reproduce in binary fission, sometimes they stay connected to each other. And when they do, based on what direction they're dividing in, you end up with a pair or a chain or a cluster. Or whatever the case may be in each of these. Well, for bacilli cells, we never see the type of binary fission that will lead to the full clustering that we see for Staphylon. So that is not an arrangement that we see there. Okay, one last thing I want to point out on this figure, just because it's worth noting, this morphology that says Coccobacillus, as you probably determined, Coccobacillus is a combination of the 2 morphologies that we talked about, both cocci cells and bacilli cells, and you'll notice that it's still elongated. This is a type of rod, so this is a type of bacillus cell. However, because it's not quite as stretched, because it's more compact like a rounder coccus cell, we call it a term in between the 2, Coccobacillus. If we take a look at the morphology and arrangements in a little bit of a different way, I'm not going to spend much more time here, but I did want to highlight that what leads to these different arrangements, as I mentioned, is how they divide in binary fission and whether they stay connected to each other or not. And here on the left side of this graphic, it actually shows how, depending on how many planes of division are present for a species of bacteria, so a cell may divide this way, and now you end up with 2. It may divide this way and this way, right? Now you can end up with 4 connected. It may divide this way, and this way. Now you end up with the 8 that are connected. So the directionality of division is what leads to these different arrangements. That and whether or not the cells will stay connected to each other. Staphylon is where you have cells that will essentially divide in many different divisions, and so in many different planes. So as a result, when they undergo binary fission, you end up with this full sort of random larger clustering effect that we see. Okay, if we take just a little bit of time to do some practice, this slide gives some real pictures of different bacterial cells so that we can identify their morphology and arrangement. And this is helpful because different species have a unique morphology and a unique arrangement to them. In other words, E. coli will always be bacilli. Staphylococcus will always be Staphylo arrangement, cocci morphology. And so it's a way that it can help us to identify what type of bacteria we're looking at. So go ahead and I recommend pausing the lecture, and we'll go through these really quickly. Try to determine the morphology and the arrangement for each of these pictures, and then I'll go through it. You can come back and listen to me talk through it. Okay, so this upper left image has these dark stained purple cells. Those are bacterial cells. And when you take a look at those, you can see that their morphology is bacilli. They are rod-shaped, they're not perfect spheres, so those are bacilli. And then in terms of arrangement, we see that they are chained together. So these are strepto. Okay, if we take a look at this upper right image, you may have noticed that the cells aren't perfect spheres, but they also don't seem quite as elongated as what we saw previously, and you'd be right. So these are bacilli cells, but we could put these in that category I talked about at the end as coccobacilli cells. They're somewhere in between. They are rod-shaped, they are bacillus. However, they are a bit more compact and a bit closer to that spherical morphology. So coccobacilli. Now, in terms of arrangement, you may be thinking, well, some of them appear to be palisades, some of them appear to be maybe diplo. One thing to keep in mind when you're looking at arrangement is we want to zoom out as much as possible and look at what do we see overall and what do we see with consistency. When we looked at that first image, we could see that every cell that we took a look at was in a chain. So that was very obviously a strepto arrangement. In this case, if we zoom way out, sure, we have what appears to be some potential groupings of cells throughout the field of view that we're looking at. However, we also have quite a few cells that are just floating on their own. And so what is likely occurring here is that the cells just happen to be clumping to each other as a result of the process of staining, something we'll do in lab, that allows us to even view the cells here. But because we see a lot of those individual cells throughout, and there's not really a defined uniform connection that all cells possess, these are singular. Now, that may feel a little bit weird. We'll get more practice with this in lab during this semester. But like I said, keep in mind you want to zoom out and really look at what do I see? And if you see quite a few cells on their own, very likely that you have cells that are just singular. If we take a look at this lower left image, we see that the cells are cocci because they're clearly more spherical in shape. And then also they're pretty obviously clustering with each other. So they're staying connected and they're forming more random clusters. They look like grapes to me. These cells are Staphylo arrangement. So this would be a Staphylococcus species or Staphylococci. Okay, and then if we take a look at this last image in the lower right, I do want to highlight for you that this is a type of cell that we will not see as often in our course, but it's still worth taking a look at an example. These are one of the spiral shapes, likely a spirochete type morphology, and their arrangement is singular because most of them are on their own with no definite connection to others. If we zoom in on pictures from real-world environments, such as the intestines or the tongue, you can see that the morphology and arrangement of the different cells still hold up here. On the right, the image from the tongue, you really see some good examples of this. Through the middle of that image, we see what obviously look like bacilli cells that are in a strepto arrangement, right? Pretty clear chain in terms of the connection, and then obvious rod shapes. You can go through and take a look at the others and practice describing morphology and arrangement. Again, it's something that we will do quite a bit in lab throughout the semester. Okay, So that is another way that we can look at cells, get some information to describe what type of cell we have, get more information about what species it is that we are looking at. From here, we want to transition into talking a bit more about some cell structures, but also their building blocks. So the molecules that we will see that go into building the cell structures or the parts of the cell that have some functionality. And in doing so, we're going to take a little bit of a tangent here. We're going to take a little bit of a side road to mention a microbe that we talked about in the first lecture that you're very likely familiar with, but that can cause some issues for us in microbiology, or at least in the way that we think about our definitions and what we've talked about before. And that microbe is a virus. Okay, now why is it that I mentioned that in terms of talking about viruses? Because you may recall back in the first lecture, I said, here is our definition of microbiology. It's the study of living things that we can't see with the unaided eye. And one of the listed microbes that we had there definitely violates this definition. And the microbe that very, very for sure violates that definition is a virus. But viruses are definitely a part of microbiology. They definitely fit here. Okay, so what is it that I'm getting at here? I want to start with this quote from Sir Peter Medawar that mentions a virus is a piece of bad news wrapped up in a protein. Viruses are quite simple in terms of their actual structure in that they are essentially two things. They are a nucleic acid, so either DNA or RNA, depending on the virus that we're looking at, and proteins. And the proteins form a package for the nucleic acid, for the DNA or RNA. And that's all that there is for a virus. And why that's important is because viruses violate our definition of microbiology because with only these two things, they are not living. They carry out a lot of the characteristics that we see organisms carry out. They reproduce, they have nucleic acids made of macromolecules, right? A lot of those things that we've seen in different areas. They can evolve and adapt over time. But crucially, they do not carry out their own metabolism. You may recall that was one of those highlighted characteristics that we said is true of all organisms back when we opened with what is life in the course. So a virus, even though it is a crucial part of microbiology and something that we will see during the semester, keep in mind that it violates most of what we've talked about so far. But viruses are a good way to introduce the idea of some macromolecules because they identify for us that there's this direct relationship between nucleic acids and proteins, which we'll talk about here in a second. But you very likely in previous biology courses talked about nucleic acids, at least DNA, but probably DNA and RNA. You've probably talked about proteins, and we want to consider how these connect. If viruses aren't living and they don't carry out their own metabolism, how is it that they're able to continue to exist? They have these two things. What's the relationship between these things and how is it crucial for life and its processes? All right, that brings me to the concept of something in biology that is known as the central dogma. Sometimes it's known as the central dogma of molecular biology or the central dogma of biology, or you may have previously learned this as gene expression. All of those are terms for the same thing. And the central dogma, I would argue, is the most important biological concept that we cover in this course and that's covered in most biology courses because it describes a fundamental process that must occur in order for life to occur. And that's because it describes the flow of information in an organism. So how are we able to actually have information lead to functions being carried out by a cell? You are probably aware, even if you haven't covered this directly, that information exists within living things. We inherit information from our parents. Information is passed from generation to generation. It's what allows for species to continue. All of our cells contain information. And you probably learned about this in terms of genetics and maybe inheritance. We'll talk somewhat about that when we get to Unit 2, but for now we want to focus on how does that information end up resulting in a living thing carrying out a physical function. And the way that it works is through this progression from the molecule of DNA to the final product of the process, which is proteins. So we have this flow of DNA goes to RNA, goes to proteins. And DNA, as you've likely learned before, is the nucleic acid that stores information or stores genes, which are recipes for how to make proteins. And proteins are a macromolecule that we all make that are the doers in the cell. These are like little machines. They carry out the jobs that need to be carried out in order for the cell to accomplish the jobs that it needs to accomplish. And so how do we have this progression from information to actual physical function? This overall flow. DNA goes to RNA, goes to protein. And I like to describe this flow in terms of an analogy to a cookbook, where DNA is like a cookbook for each of our cells. Imagine you have a family cookbook that has been passed down for generations, let's even say thousands of years, that has the best recipes that you can think of. And you really want to make sure that you keep that cookbook in good shape so that you can continue to pass that down to other members of your family in the future, and you can keep doing it. Okay, well, if DNA is a cookbook with recipes for how to make certain meals, where is maybe the last place you would want to take that DNA if you were trying to prevent it from taking any damage? I would argue you may want to keep it out of the kitchen, because in the kitchen it could rip, you could have spills, you could light it on fire accidentally, and that would damage it to the point of not being able to pass that information on to other members of your family. So if you wanted to make a meal out of that cookbook, but you didn't want to bring the cookbook in the kitchen, what could you do instead? And probably the easiest solution is you could go to whatever recipe it is that you wanted to make, and you could rewrite that recipe in a temporary form. Write it on a piece of paper, copy the information down. You still have all of the same information, but now you don't have to worry about the cookbook taking damage in the kitchen. Well, that's what our cells do as well. We have this pristine, this perfect, this untouched version of recipes for how to make individual proteins for the cell to be able to carry out jobs that are all in the DNA. And in order for the cell of being able to avoid that DNA from taking damage, it's going to make a temporary copy of the recipes that it wants to make. So the DNA gets rewritten as another nucleic acid called RNA. And that RNA is just a temporary copy that's going to carry the information, carry the recipes to the ribosome, which is the part of the cell where proteins end up being made. And once the proteins are made, the recipe is complete, those proteins can then go to wherever they need to and carry out whatever jobs need to be carried out for the cell to do what it needs to do. This is fundamental to life. We have information storage, information delivery, and finally, the actual physical function that results from that information being turned into protein. Okay, if we take an even deeper dive into proteins, there are some characteristics about them that are important to understand so that we know where the physicality of this whole process occurs. Proteins are made up of small building blocks known as amino acids. You've likely seen this before, but each amino acid has three groups that are all identical to each other, known as the backbone. We have the amine group shown here in blue. We have the carboxyl group shown here in yellow. We have what's known as the alpha carbon linking the two together, which has its own hydrogen attached. And then coming off of the alpha carbon, we have what is known as the R group or the variable group. And each different amino acid has a different R group or variable group that gives it different properties that allows it to help carry out different jobs or move the protein in different ways. There are 20 different amino acids that occur naturally. Each of them has a unique R group, giving it a different characteristic, which makes it behave within the overall shape of the protein differently. Sometimes you will also hear that variable group or R group described as the side chain. No, those are all just terms for the same thing. And we'll take a little bit of a deeper dive, but we'll pretty much just end there for our course. Now, in order for a protein to be made, we have to take multiple amino acids and we need to chain them together in a very specific sequence. That's what our genes or our recipes code for in terms of how to make a protein. So a gene will tell us first you put in this amino acid, next this amino acid, next this amino acid, and so on. And those amino acids get added to each other in that specific order. They do so through the formation of what's known as a peptide bond, which is a really strong bond that will hold the amino acids together permanently in that order. And that formation of the peptide bond is carried out by what's known as a dehydration or condensation reaction. Water is lost, the two groups become really reactive, so they stick to each other to become happy, and in the end, we end up with these chained amino acids in a specific order. And the order of those amino acids and how they are connected to each other is what we call the primary structure of the protein. And fundamentally, the primary structure is really, really important for the protein in terms of how it's made, but it's really not that important in terms of how the protein actually works. And that can be a little bit of a weird thing to wrap your head around. So I want to walk you through the ways that we describe the overall shape or structure of a protein. First, we have that primary sequence, which is the order of the amino acids connected by peptide bonds. You can see here Phi, Gly, Arg, Cis, Glu, Lu, Alla, Alla. They're in a specific order and they're connected to each other, essentially in that straight line form. From there, though, the amino acids aren't happy being in that straight line form. They actually want to interact with each other. And so they will start to twist and turn on each other or zigzag back and forth in space to have interactions with other amino acids that they get along well with. And in doing so, they form what's known as the secondary structure. And secondary structure of a protein still isn't that important for what it's going to do overall in terms of its function, but it is starting to develop a little bit more texture, right? Instead of just a straight line of amino acids in a chain, now we have a little bit more shape to it. Texture here and there. We have twists, which we call alpha helices, and we have sort of zigzag sheets that form that are known as beta pleated sheets. And all of these secondary structures are held in place by hydrogen bonding and start to make the amino acids more happy in terms of where they exist in space. However, what's truly important for a protein to be able to have some specific job, to carry out some specific function, is its overall three-dimensional shape. And when I say shape, that's probably even what pops into your head, right? If I say, what is the shape of a ball? You probably don't think about the material flattened out. You probably think of it's a round sphere. And what we're actually describing there, the three-dimensional shape, is what's known as tertiary structure. So something to keep in mind is what is the most important thing about any protein in order for it to be able to do its job? It's the shape. It's the tertiary structure. And the tertiary structure forms because in order for those amino acids to become even more happy, they start to ball up. So you can imagine we have amino acids in a line, they start to twist and turn or zigzag on each other to form secondary structure. And then those secondary structures ball up. And now we end up with some specific three-dimensional shape that determines what the protein can actually do. Okay, from there we have what's known as quaternary structure, which is something that we use to describe still three-dimensional shape, but for those proteins that have multiple subunits or multiple chains of amino acids. So about 2/3 of proteins are estimated to contain more than one chain of amino acids. So you can think of it as multiple big pieces coming together to reach the final form of the protein that we call quaternary structure. If a protein has two subunits, how those two subunits align with each other and are held in place is its quaternary structure. If it has three subunits, how those subunits are held in place, quaternary structure. Four subunits, same thing, and so on and so forth. For the 1/3 of proteins approximately that do not have multiple subunits, I want you to take a second and think about it. What is their highest level of protein structure? The answer, as you probably came up with, is tertiary structure. So if a protein only has one subunit, it does not have quaternary structure. Once it balls up into its three-dimensional structure, that is its overall shape. Tertiary structure is the highest level of structure that we can describe the shape of that protein in. Okay, the importance of the R groups that stick out that we talked about back from each amino acid that makes each amino acid unique is that those R groups have characteristics. So they could be positively charged or negatively charged. They could be hydrophilic and love water. They could be hydrophobic and hate water. Those characteristics of the side chains are what drive the overall folding of the protein into its three-dimensional shape. So the tertiary structure and quaternary structure, if we're talking about a protein with multiple subunits, is ultimately determined by how these R groups want to interact with each other and hold the protein in different spots to allow for it to fold into its overall 3D shape. Some R groups end up being free from interacting with any others. They can be important for carrying out chemical reactions with other molecules in the environment. But largely it's these R groups or these side chains, these variable groups in amino acids that ultimately result in the function of the protein. They bring together the protein into that ball for tertiary structure. They can allow subunits to stick to each other for quaternary structure. And ultimately they can interact with their surroundings to carry out whatever jobs need to be carried out that the protein is actually accomplishing. And you can see here just another image. There's a video there if you'd like to navigate to it, if you want to take a look at proteins and protein folding. And then this slide is just essentially highlighting what I mentioned, that R groups interact with the environmental molecules in order to carry out jobs. In this case, we're talking about an R group binding to a substrate, so holding on to something that's going to be involved in a chemical reaction and positioning it just perfectly so that that reaction will actually take place. Again, a couple links to YouTube videos there if you want to make note of them. And watch how this folding and how this holding takes place for proteins to carry out their jobs. Also important to proteins, although not something we'll spend a lot of time on, is that how they fold up into their three-dimensional shape results in different pockets or different areas that water can go into or that water cannot go into. And that can be really important for specific chemical reactions to take place because certain molecules will be carried in with the water. Sometimes water itself is involved in those reactions. So water availability and water accessibility to different regions of the shape of a protein can be really, really important for both how it folds up, but then also how that shape that it folds up into allows for the protein to actually do its job. If we take a look at this picture down in the lower left associated with one of these videos, the one thing I want to highlight for you is here's an example of a protein called an enzyme, which we will talk about in here. And you'll notice that there's one thing that likely stands out, and this is a cartoon, so it's not a real image of a protein, But one thing should really stand out to you about that shape that we're looking at, and that it is uniquely in 3 dimensions, a shape that is probably going to do something, right? Certain things can fit in here. The shape really stands out as unique. And that's because that's the most important thing about that protein. And then we'll just leave that for now. Okay, did I mention that protein shape is everything? Of course I did. But one last thing that I want to mention is that it is so important that we can even see it through through a process known as denaturation, and then if we're able to put a protein through the reverse process known as renaturation. Okay, if we take a look at just this flow chart of proteins in the upper right, you'll see that on the far left is an active protein. So that is its normal shape. That is the shape that allows it to do its job. If we expose that protein to changes in temperature or changes in pH, especially, the protein will unfold. Those changes will affect the environment. As a result, the protein will wiggle around in different ways and no longer hold its shape, and we see a denatured protein. And a denatured protein, since it's not holding its shape, won't carry out its function. Okay, but importantly, if we put that denatured protein back into standard conditions, or the conditions that it was originally in, what we will often see is that it will be able to refold back into its original shape. And if it is able to do that, we will see that it will have function again. So we have a really clean way to demonstrate that protein shape really is everything. This AlphaFold protein structure database that you see here is associated with a recent Nobel Prize, and how proteins fold into these specific shapes. It's something that is now largely predicted using computers. I definitely encourage you to take a look at it if it's something that you're interested in. It's actually pretty cool how it works. But we have a huge database that allows us to both predict and also see proteins that have been visualized before, so we can determine what their shape really is, and how that plays into their function, how interactions occur, and if we make changes to that shape, how the protein will no longer work or work to a lesser degree. Okay, so bringing that back, examples of cell structures. Last lecture, we talked about a variety of different cell structures. Examples of molecules would be things like the nucleic acids and proteins that we've talked about so far. We'll talk about some other molecules as well, but keep in mind that it's molecules that go together to build cell structures, and in either case, you can be talking about a virulence factor if it's something that is helping a microbe to cause disease. So just a little bit of a review to connect those physical things we've now been talking about back to the concept of virulence factors, and how these can play a role in human health overall. I want to give a really clean example where we circle back and talk about something more specifically that was mentioned in the last lecture, which is the glycocalyx. You may recall that when we talked about the glycocalyx for prokaryotes, we talked about how there were 2 versions, the slime layer or the capsule. And I want to really spend some time focusing on the capsule, because the capsule is a cell structure that is made up of the molecules, glycoproteins and glycolipids, and capsules are absolutely virulence factors. Okay, why is that the case? And when we think about all of this organization and what we're talking about, how is all of this connected? When we take a look at a specific capsule, we have a cell structure that is doing something that is helping a bacterial cell to ultimately cause disease. And that's because capsules actually have 4 different ways that they carry out a job that results in it being harder for us to get rid of the bacteria. Capsules help bacteria to attach to surfaces, making it harder for them to just be washed away or go somewhere else. You can see this especially in the lungs or within wounds. They also protect against macrophage digestion, which is something we'll focus on when we cover immunology in Unit 4. But for now, you can think of this as a type of camouflage from our immune system. That was a key part of a virulence action that could take place, right? They also protect against desiccation. So this is something that's a little bit more just a standalone benefit as a cell structure for a bacterial cell, but they're able to help retain moisture within that cell so it doesn't dry out and die. And they can block or inhibit some antibiotics from getting through, which makes it harder for us to treat certain bacteria that act as pathogens. So you can see here that capsules provide, as a cell structure, a lot of specific benefits to a bacterial cell, making it better at infection and harder for us to deal with. So when we think about it all together, we've got glycoproteins and glycolipids building this cell structure. The way those things act with each other, they form a barrier-like glycocalyx that can attach to surfaces, that can serve as camouflage, that can prevent drying out, that can block antibiotics. As a result, we have a virulence factor that is really something that assists these bacterial cells in causing disease. And in fact, if we take a look at two very well-known species that can have a capsule, Streptococcus pneumoniae, and Neisseria meningitidis, I always can't say that, but as you guessed, it causes meningitis, and Streptococcus pneumoniae, which causes pneumonia. Both of these are really, really tricky organisms to deal with for the reasons stated before. They have capsules, they have this cell structure that gives them added protection and also increased ability to colonize us in areas where we obviously don't want them. So it enhances these organisms' ability to cause disease and act as a pathogen. And in fact, as a general rule, if you have a bacterial cell that has a capsule, it is very, very likely that it is a pathogen. It is very likely that our body will have trouble with it, and so therefore it becomes very likely able to cause disease. That doesn't mean that it's necessarily going to kill you or cause very, very significant disease, but it is very likely a pathogen if it has a capsule. We'll talk more about Griffith that's mentioned down at the bottom in a future unit. Okay, another type of structure that we see that actually develops outside of cells, but is still a virulence factor, is something that is known as a biofilm. Most microbes that are just living in the natural world will exist in a biofilm because it gives them strength in numbers and it allows for them to form a community of sorts that ends up being really, really beneficial. A really good example of a biofilm that you are likely familiar with is dental plaque. So that slime that forms on teeth, and now everyone just licked their teeth. But that film that you can feel is a collection of different microbes that are creating what's known as a biofilm. And the way that biofilm development works is you have an initial stage where some microbe attaches to a surface. This is crucial, and you'll learn in this course that attachment is a physical step that occurs very early in a lot of processes that really matters. Why? Because a microbe has to be wherever it is that we say something is occurring, right? And wherever that thing is occurring, not just where we say. So a microbe has to attach to a surface, and then from there anchor itself and start to release molecules that form what's known as an extracellular matrix. Now, this extracellular matrix is generally a collection of DNA, sugars, some proteins, some lipids, but a lot of just sort of stuff, a lot of molecules that cells will release in order to create a sticky-like substance. That can assist other cells in attaching to the area, and then also it provides sort of a protective layer, as we'll see, that helps to keep all of the individual microbes that exist within the biofilm safe from things that are passing in the environment. Over time, you'll have maturation of the biofilm, both because other microbes start to deposit themselves within the biofilm and replicate, and then also the initial microbes in the biofilm reproduce. So you get this increase in numbers. You get an overall larger size. So a slimy-like film starts to form on a surface. And in doing so, you also get the development of water channels so that nutrients can pass through all of the different microbes, and water and those nutrients can be delivered to all of the different cells that exist within the layers. You can imagine that as the cells increase in number, just like if you're in a crowd, everything starts to fill up more and more and more. There has to be routes for those materials to get to all of the cells to stay alive. Finally, though, if everything is really, really, really crowded, the cells will start to run out of nutrition and space, and some of them will break away. It doesn't make sense for them to stay there. Even if a biofilm offers protection for those individual cells, they're going to die if they can't get that nutrition or if they don't have space to replicate. So some of them will break off, they will flow to another area where they can attach and make their own biofilm in that area. So biofilms provide some protection, but ultimately if they get so big, the cells within them even have to move to a different location to start a new biofilm. Okay, so how do bacteria know to form a biofilm? Well, there's a process that they carry out that is known as quorum sensing. Quorum sensing is where molecules known as autoinducers are made by each cell to communicate to others in the area to say, hey, I'm here. Okay, and why does that actually matter? If you think about it, if one cell attaches to a surface and says, okay, I need to make a biofilm, it could make sense, but that cell's taking on a lot of risk. Because if there are no other cells in the area to help it to make the biofilm, that cell risks dying, essentially killing itself from working too hard. If all of its energy goes to making that extracellular matrix, so all of those materials, the proteins and the DNA and the lipids and the sugars that it needs to create the slime surrounding it, if no one else ever comes along, then it just wastes all of its energy and it can't carry out the other jobs that the cell needs to accomplish. So quorum sensing is a way for cells to identify that there are other cells within a close proximity. And the way this works is that all cells will always be putting out molecules known as autoinducers to their environment. It's just sort of a standard signal that they send out to their local environment. If another cell comes along, it may detect, hey, there's an autoinducer, there's another cell in this area. It makes sense for me to stay here, attach, replicate, and start to build something. Well, now if both of those cells are there and they're both releasing autoinducers, there's more and more autoinducers in the area. So now it becomes even more likely that another cell that comes along detects that there are cells in the area. And if it does, it may deposit itself in that area and attach and replicate. And over time, we get this buildup of more and more cells creating autoinducers to signal, hey, we're all here. It's not just one of us, there's a bunch of us here. And importantly, this overall system also has what's known as positive feedback. As that concentration of autoinducers gets higher and higher and higher, and the cells that are in the area detect, hey, there's more and more cells here, it makes sense for me to form a biofilm. I'll be sharing the job. I don't have to spend all of my energy. We'll all just spend a portion of energy. As that signal gets stronger and stronger and stronger, gene expression changes. We talk about gene expression, central dogma. And these cells turn on making the autoinducers to an even higher level. So now they release even more of them to give a stronger signal. So over time, as you get this biofilm formation and you get these numbers of cells building up in a space, you get a stronger and stronger signal telling other cells, hey, I'm not alone. There's a bunch of us right here. You should join to help. We'll all share the burden. We'll all share the energy cost, and we can together create a successful biofilm. Okay, now the benefit of a biofilm, there's a variety of things that can be beneficial about a biofilm. However, what really is important is that we get layers of cells along with an extracellular matrix that allows for protection from things that could affect those cells. Oftentimes, this could be antibiotics, it could be immune cells in some cases, but largely anything that could be toxic or harmful to the cells within this biofilm, they are receiving protection from it because there are so many of them. Even if some of them get wiped out, which we expect would happen, those that are down at the bottom or in the initial stages of the biofilm or in a protected environment are able to survive the dosing of those drugs because they never have to come in contact with it. So we'll talk more about this in the future, but here's another way that microbes can have a virulence factor or biofilm that's forming in order to be tougher to deal with in terms of causing disease. And again, something in line with these molecules that we've been talking about and these structures that get built in order for it to be organized and have specific functions, in this case, leading to potential disease. Okay, here is an image of some of the different exterior structures that we've talked about. Over here on the left, we have some staining that reveals capsules. So you can see the cells on the inside that are stained. These look like cocci cells that are in Streptos. This is Streptococcus with a clearly defined capsule surrounding each of them, obviously well connected and highly organized. Up here in the upper right, we can see that there is a biofilm that we're taking a look at. In this case, it has some yeast that has been deposited within the biofilm as well as bacteria. And you can see that between all of these cells is the extracellular matrix that makes up that slimy protective layer. A big reason why we care about biofilms in healthcare is because catheters can very easily develop biofilms and can lead to infections spreading. Think about it, when the biofilm builds up, some of those cells end up leaving because they don't have enough space and nutrition. They can leave and get to some really difficult areas of the body that are tough to treat. So you can end up having an infection spread as a result of biofilm formation on this artificial surface. So it's something that we have to be acutely aware of in healthcare so that we're making sure to avoid the potential for that issue. All right, we're going to go ahead and call it here for this lecture. The next lecture will continue talking about a cell structure that we focus on quite a bit in this course, but it'll be a shorter lecture that focuses on just that structure, and that is the bacterial cell wall. So I hope you have a great rest of your day, and I will see you in that lecture.